Hi, everyone. Welcome. I am Lindsay Jibb. I'm a scientist here at the Sick Kids Research Institute. Really my great pleasure to be able to introduce Dr. Salter today. Dr. Salter has a very esteemed background. So Dr. Salter is a senior scientist in the neurosciences and mental health program at the Sick Kids Research Institute and a professor in the Department of Physiology at the University of Toronto. He completed his term as Chief of Research and now holds Chief of Research Emeritus within the Sick Kids Research Institute. He received his medical doctorate at the University of Western Ontario and his PhD from McGill University. Dr. Salter determines fundamental molecular and cellular mechanisms of normal and pathological neuroplasticity. His discoveries have broad implications for the control of cell-cell communication throughout the nervous system, and his work has regularly appeared in elite journals, including Nature, Science, Cell, Nature Medicine, and Neuron. He is using his discoveries to design and test new types of treatment for individuals suffering from a variety of disorders of the CNS. He is developing molecules that target major cell signaling pathways in neurons and in glial cells involved in pain, stroke, neurodegenerative diseases, and schizophrenia. He has won numerous awards for his work, including an international research scholarship from the Howard Hughes Medical Institute, and is a fellow of the Royal Society of Canada. Please join me in welcoming Dr. Salter.
Thank you, Lindsay, and thank you to Fiona and the organizers for the chance to talk here in my home institution for once. So I was listening to the lecture today and the questions and trying to think, why would most of you even stay around for my lecture, let alone be interested in it? Now, I know Fiona, she's just a nice person and, just is here to be supportive. I'll try to give you a sense of why the things we're doing might be relevant to the things that you're doing, most of you. The people in the back are people from my lab and, they know what we're doing. So this is the kind of situation we're all interested in. We're here for this reason, and we've been hearing stories about this all day. So to try to make this relevant to you, I harken back to what Massey was talking about today, about how brain circuits are modified. So, the aphorism that pain is in the brain is actually true. I do believe that, right? It is true. So all of these things that we're doing, I'm going to tell you about neuroimmune interactions, and Hannah told you a little bit about that. And we heard about culture and ethnicity and gender and sex and all these things that can influence. They all get together and they influence the way the structures and circuits in your brain work. That's how all of this happens, right? All of these things that we talk about in the biopsychosocial model, they feed through and change brain activity, right? If that's not true, modern neuroscience has failed dismally, and you have to come up with an alternative. Okay, so I'm going to start in and just tell you a little bit about where the stories that I'm going to tell you came from and how it might be useful to think about it this way. So this is going to go back 22, 3, 4 years or so. I guess you can tell when the speaker's getting old because they harken back, to the good old days when life was easy and things. So this part of the story is one that you're going to see repeated in this. And if you were paying attention, you might have heard a little bit about this from Hannah's. And the idea was from a really good friend of mine, Yves de Koninck's lab, that normally there is a lot of inhibition in the circuits that send nociceptive information up into your brain, right? And a lot of that signaling and circuitry is in the spinal cord here. And he had come up with a kind of funky mechanism about this, at least a little different at the time, that the inhibition is mediated largely by GABA and glycine there. And for this inhibition to be powerful and good inhibition in the adult nervous system, then one needs to have the activity of this pump working well to keep chloride low. And that's all I'm going to say about that, okay? The idea was that dysfunction of this pump could then allow chloride levels to rise. Inhibition wouldn't be nearly as powerful as it would be. And so the filtering event that keeps pain signals going, nociceptive signal transmission in the spinal cord down normally gets lost, and there's an amplification of the signaling up into those networks that Massey was telling you about in the forebrain, in the amygdala, all those areas of the brain, okay? So this was an interesting idea. The week before this in Nature, we had published something that started essentially with what Hannah showed. So I'm going to tell you about the importance of microglia, even though Hannah just told you microglia may not be important, right? And you'll see why in a second. So this was an observation we had made with a colleague in Japan, Kazuhide Inoue, and his graduate student, Makoto Suda. And at the time, the world, like Eve and everybody else and me before we did this, everyone was concerned with neurons, and how did the nervous system work, which meant how do neurons work? And we had this observation that injury somewhere a couple kilometers over there, not in the spinal cord, but changes the reactivity or the state somehow of a specific kind of cell type, the microglia. And this is something that, some of us knew a little bit about, but mostly we didn't care because we were obsessed with neurons. We were generally, as a field, we were ignoring this. And personally, as me, I was ignoring this until Makoto had done an experiment like this, where he used this drug, TNP-ATP, in a model like the one that Hannah told you about, where you get hypersensitivity. That hypersensitivity is reversed by a spinal or intrathecal injection of this molecule, which we knew was a blocker of a specific set of receptors called P2X4 receptors. So these are receptors that are normally activated by extracellular ATP. And we went through a number of different molecular biological tricks to ascertain that was really P2X4 receptors. And so one of the questions we had was, which cells are the P2X4 receptors? And, like my bet was fully on neurons. Once again, my lab people will tell you I was wrong. And it turned out it wasn't neurons. It wasn't the most popular glia type at the time, astrocytes. It was microglia. And so we knew right away from this observation that, as much as I hated to admit it, because P2X4 receptors are on neurons, P2X4 receptors are necessary to maintain the hypersensitivity. P2X4 receptors are microglia. The microglia must be maintaining the hypersensitivity. There was no logical way around that. Okay, so then this is one of these observations, maybe some of you will have them in your lifetime, where my worldview changed like that, right? It's like, man, there's more than neurons. What are we going to do? So it turned out a couple of weeks after this, I met up with Eve at a meeting, and we both, I think, said the same thing. Hey, maybe your story is my story and my story is your story. So we got together and had a couple really dedicated people in each of our labs, Jeff Kuhl in Eve's lab and Simon Biggs in my lab. And we came up with this concept that has been really driving my thinking about this area of spinal cord gating of sensory information ever since. It's the basis for this little, well, you might think of it as a delusion. I call it a conceptual framework. So this is the idea that after a nerve injury, then, or other things that I'll tell you about in a second, the microglia respond. We saw they change their number and shape and everything like that, and that they signal to downregulate this potassium chloride cotransporter by release of this peptide called brain-derived neurotrophic factor. Okay, so this is a simple little model here, right? And that idea of loss of inhibition has been key to what we've been doing ever since, right? And in this case, it's thinking about how do microglia come about to do that. So that was, 19 years ago, 20 years ago or so. So we've been spending a lot of time working out the molecular and cellular details behind all this and trying to understand what's going on. And as we go along, this fundamental model gets more and more complicated because we add more and more stuff to the middle of it, right? So we go and we figure out how P2X4 receptors, which are not normally expressed in microglia, how they get upregulated. How does BDNF get released? What does BDNF act on? How does that cause downregulation of KTC2? And oh, by the way, along the way, we discovered that NMDA receptor function, so excitation, is accentuated. So there's not just loss of inhibition, there's facilitation of excitation. Right. So it's like, if Fiona was driving her Porsche along the 40, the Lamborghini that she has, she's driving it on the 407. It's the only place you can drive fast, right? So she's driving it along there and she's doing what my driver's ed teacher told me never to do. To maintain a constant speed, you can put your foot on the accelerator and on the brake, right? And if you do that until the Lambo's brakes burn out, you're going to be able to maintain a nice speed. You can go faster by pushing more on the accelerator or releasing the brake a little bit. And it's both of these things that we think are going on. Okay, so this is the model, right? And the model had gone on like this happily, working out more and more details about minutia that are related to that and how it fits in. So we figured out over the years, how does this fit into inflammatory pain as well as nerve injury induced pain? And how does morphine induced hypersensitivity fit into this kind of framework? Now, one thing that we didn't count on, at least I didn't count on, and once again, I was wrong, didn't count on, was that this model, this concept, at least on this side over here, which you could think of as the glial or immune side, this side had all been worked out in only male animals, right? So, 20 years ago, only every experiment, all these papers, dozens of papers, not just from our labs, but other labs, everyone's working only on male animals. And then it took a little bit of a shock for us to do some experiments prompted on by our good dear friend, Jeff Mogul, and to figure out that this is only the male side in rodents. And something happens in females where this part of the entire pathway doesn't get activated. The microglia still respond somehow, but the P38 MAP kinase, all this stuff that causes release of BDNF doesn't occur, but yet there's still signaling over here, right? And Hannah pointed that out to you in the microglia independent. So this is a microglia independent kind of pain hypersensitivity initiated by nerve injury that doesn't require microglia. So Hannah showed you one kind, this is another kind in female rats and mice. Okay, we're still working through that to try to understand what's the basis for this line here. So we've got that, and then we've been working on more stuff over here, NMDA receptor subtype, other enzymes, lots of things going on over there. So that's kind of where we were until we started down this story that Hannah told you. And really the place we're at now is that, we've got a number of ways of activating this side on the immune side in the spinal cord. And then we've got ways now of activating this on the immune side in this microglia independent form, but there's a convergence in the middle here. Okay, so this is the real underlying theme that you're going to see one more time in a couple minutes. Okay, so this has been the model. So we start off with this what at the time seemed complicated, but now we think of as a relatively simple model. And we've just been gilding the lily and figuring out different kinds of things that can affect or inhibit in the model. Okay, so remember this, we've got neuroimmune signaling in a number of different ways. This is, Alan Basbaum gave a talk at the ISP meeting and about what's changed in 50 years in the field of pain. And Alan said, yeah, basically nothing, except the work in his lab, of course, but nothing really had changed. And it's like, dude, 50 years ago, no one cared about neuroimmune signaling. It wasn't even a thing back then. And now every meeting you go to, somebody talks about neuroimmune signaling and pain. So, once again, Alan is Alan. So, okay. So enough of that. That was more an inside joke for the few ISP people. Okay, so in the rest of the time, I'm going to tell you two short stories. One about yet another kind of neuroimmune signaling. And the last one is to try to break out of this in a completely different way. Okay, so this one is going to be really fast. This is the PhD work of Mashad, who graduated a few months ago. And she was interested in immune cells in the meninges, right? And so for those of you that don't think anatomically, which is often me, the meninges surround the central nervous system, not just the brain, but also the brainstem and the spinal cord. And within the meningeal spaces, there are lots of cells. And there are lots of immune cells, and they come in essentially all the various flavors of innate and adaptive immune cells that you could imagine, living in the dura or between the arachnoid and the pia, and getting out in there and doing stuff that we're only really starting to, we as a community, not we, but that the community is really only starting to understand a little bit the importance of meningeal cells and intrameningeal and dural cells in a whole variety of disease processes and in the normal biology of the central nervous system, right? And so Masha was interested in specifically one type of macrophage. You heard about macrophages from Hannah, not in the peripheral nerve, but in the meninges. And this is a type of macrophage that expresses this protein called the CD206 mannose receptor. And so I'm not going to show you all the data because you're not going to remember it anyway. And the only people that are going to criticize it are the people in my lab, right? So the upshot of this was related, and funny thing for people who, for students, this actually came about because of a failed control experiment, basically, right? And so here we end up now, most models have, pain models in rodents have some big injury. This is a minor injury producing hypersensitivity. And that was because the minor injury is the one that we use for sham in a lot of experiments. And basically, Mashad found out that these CD206 positive meningeal macrophages are required to suppress pain from going all over the place. So when she knocked those off in various ways, then she saw more widespread pain hypersensitivity than you'd get by a small injury. And the injury that we would typically use in these kinds of, models of, traumatic nerve injury and things like that are a small incision in the thigh just to get at the sciatic nerve. Okay, so that's the essence of the story. And you're going to see a little bit about that because I will show you a couple of data slides related to that. Because remember, Fiona, I told you had to remember this side of the diagram, right? So this is the neuron part, right? That now is in the middle of our delusional conceptual framework that has your Lamborghini's brakes and accelerator. And so if we just try to simplify that in some ways, we've got the two key players, NMDA receptors and KCC2. So downregulation of KCC2 suppresses inhibition, and facilitation of NMDA receptors promotes excitation. And what Hannah brilliantly put on her slide is that there's lots of interactions here. So this is a virtuous cycle or a invirtuous cycle that you can enter either from the NMDA receptor side or the KCC2 side, and then they feed forward, okay? So we were wondering about this hypersensitivity caused by minor injury and loss of CD206 positive macrophages. And so we used these drugs that, if you've been paying attention, I know you would remember CLP facilitates KCC2 and APV suppresses NMDA receptor function, and 2 data slides. So here you can see this sensitivity produced. And interestingly, this only happens in male animals. So after the minor injury, Mashad knocks off the CD206 positive meningeal macrophages. There's this sensitization of the animals. And then in this case, she gave APV, reversing the hypersensitivity, blocking NMDA receptors. So NMDA receptors are necessary for maintaining the hypersensitivity. And then CLP290, which again reversed the hypersensitivity. So again, a different cohort of animals, all male. You see this, no hypersensitivity in females. We still don't understand why that is with this approach, but reversed by CLP290. So the downregulation of KCC2 is required. So then we're building out our model a little bit more. I'm sure you can see where this is going, right? So now we add another player, immune players. So we've got neurons in the middle again, and then we've got immune cells here and immune cells here, and now immune cells here. So these ones are facilitating microglia. These are probably a subset of T cells. These are macrophages of some sort. They're all facilitating this feed forward vicious cycle, right? NMDA up, KCC2 down. Meningeal macrophages seem to be doing the opposite. So one of the areas we're trying to figure out now is how does this happen? What is it about these CD206 positive meningeal macrophages that are able to downregulate this vicious cycle, right? So that might be one way of really thinking differently about how can we intervene to do this. For a person like me who thinks of molecules and genes and proteins and cells, immediately we go to drugs as a way, molecules as a way to do this. But if you think about it, how do social impacts come? Maybe they're changing these cells. Immune cells are very sensitive to environmental influences, right? I know almost nothing about immunology, but I do know there's lots of cells and they work in very complicated ways, and there's lots of ways to modify them. Okay, so maybe that's the key, is that it's neurons that are giving us the experience. It's neurons that are causing the behavior, right? We use paw withdrawal or escape or movement or something like this, a motor output. That has to be neurons, right? Immune cells can't do that. Microglia can't do that. But the immune cells can modify those responses that we can see in rodents, and presumably they're experiencing in their brain as well. Okay, so that's my little proselytizing about that. Now, this is the other side of things, trying to think about not about how environment and socioeconomic status and all these things could influence immune signaling in the brain, but a very targeted attempt to try to find molecules that might help that we wouldn't find in any other way. And this is from Shahrzad Ghazasieri, who came to my lab thanks to Donald Trump. That's all I'm going to say. So you need to remember what is the central dogma of molecular biology. You've got DNA turns into, gets changed and produces RNA, and RNAs encode proteins, okay? So this is the essence of all of cell biology. Sorry, Jazz, cell death is in there somewhere, but I don't know about that. The idea is that gives us a way of trying to understand what's going on inside cells by looking at changes in DNA or in RNA or in proteins. And one of the technologically relatively easy ways to do this is by transcriptomic analysis, just because the technology for DNA and RNA really has outstripped and grown much faster than proteomics. As a physiologist, I'm interested in proteins, but for this part of what we did, okay, we'll look at RNA and see if that gets us anywhere. So for full and complete disclosure, I think people in my lab knew, but I didn't know that Shahrzad had this in her mind. This was a thought experiment that we were going through, that this was a way to find drugs. She didn't tell me that until we were like 80% of the way through the experiment. What the espoused purpose of this was because Shahrzad came to the lab with a background in sex differences. And we had just discovered the sex difference along with Jeff Mogul. So it's like, oh, okay, this is now cool. So the idea might be if we can find genes or proteins that are changed in both, and we knew the sex difference occurred in rats and mice as well, right? So maybe we can use transcriptomics to find genes and molecules that are changed in both rats and mice in a sex-dependent way, right? So it was a very targeted look for finding sex-different species conserved, because in the back of our minds was the idea that if we can do that and find things that are conserved from rats and mice, which many of you think are like this close in species, but they're kind of far apart actually, maybe the genes and proteins that we would find could extend up to humans, okay? That was the idea. Okay, so she designed this very complicated set of experiments. I think in the end, she recruited everyone in the lab to help her on this because it was very complicated getting all the blinding done and getting all the groups. And so in this design, this is the group. So she used at the time this traumatic nerve injury as a way to get access to the changes that might be going on. And then we had other groups that we thought of as comparators. So, and the idea was to have lots of control groups. I try to insist in the lab on like one experimental group, 3, 4, 5. I drive people crazy in the lab about this, but the controls are in fact more important than the experimental ones. So are the control groups in this experiment actually controls that we can use as a way? And the idea was to really bump up the statistical power by having lots of control groups. Okay, so the first way Shahrzad looked at that was looking at doing principal component analysis, and you can probably see there's kind of this group where all these triangles are the experimental group, the SNI group, and these are the putative at the time control groups. So you can see that in rats and mice. This PCA analysis gives this visual impression that you can make look really good if you change the colors a little bit, you know. That's on the right track, okay? So then she looked group by group to compare all the groups together at a transcriptomics level and produce these things, which are called volcano plots. Now, I don't know about you, but they do not look like volcanoes to me, right? They are, in fact, the most boring set of volcano plots you will ever see because they are flat. There are no differences. So the control groups are not different from each other, and they're very different, as you can see much more easily here in this heat map using non-hierarchical cluster analysis. So there's a main cluster that all of these are the SNI groups, and all of these are the control groups put together, okay? So did a lot of statistical work to say, okay, the control groups are actually controls, they're homogeneous, we can use them in this to compare to the experimental group. Okay, so then we start to do that. Shahrzad did all this, of course. So now you see volcano plots the way you might have seen them before, right? You've got gene expression that's changed up compared, and lots of genes. Each dot is a gene transcript of which is changed. And this side is upregulated. This side would be downregulated in SNI versus the control groups. Okay, so lots in males and females in mice. There's a really nice correlation between the males and females. So, but it's not a perfect line, because if it was a perfect line, then we would, I guess, stop this experiment. Nothing in biology is perfect like that anyway, but there's enough variability that we're convinced there's a good correlation, but it's not absolutely 100.000% perfect. Okay. Same thing in rats, lots of genes that have changed in males and in females. So we're thinking, okay, now we're on the right track. We got this sex different, species conserved, nailed, and then you've got, again, really good correlation in rats and mice, and okay, we're coming along, perfect. So then you get faced with this kind of thing, which is, I'm sure all of you are very used to looking at 2x2 Venn diagrams. For me, it took like 12 times for Shahrzad to explain this to me about how this worked. So we're looking for sex different, species conserved. So where are those cells? Anybody got any idea? I'll help you. Here, right there, and right there. So lots of genes that are changed, but only 4 in total in the right cells. And it's like, oh man, what's going on? But then look at these right here, staring us in the face. This group, right, of 93 genes that are sex conserved and species conserved. So we had actually found something we were not looking for. Okay, this is when it got really interesting, because I like it actually when things don't work out according to your idea. Sounds kind of weird, but disproving your own favorite idea is probably one of the best things we can do in science. Okay, so Shahrzad taught herself a lot of network analysis as part of this, and so I'm going to take you through how we get to there, which I think this is the cleverest part of the set of experiments that we're done. Okay, so you can take these, you can take the genes and then run them through a variety of different gene network analyses when you look at the proteins that are encoded by the genes, and you can make these fancy schmancy, Fiona, that is a scientific term, fancy schmancy, diagrams like this, where the size of the circle is related to the influence and the interactions with all the other genes and proteins. Those of you that are counting, and I know some of you are, there aren't 93 on there because some of them fall outside this, right? This is the main group. So then for each of these hubs or genes, then you can calculate something called the integrated value of influence. Now, this apparently is pretty standard fare if you do gene network analysis, these kinds of things, these hub diagrams and these integrated value of influence. The clever part was what Shahrzad invented was to take this integrated value of influence, convolve it with information in this publicly accessible database of drug-gene interactions called the Drug-Gene Interaction Database, which has about 40,000 genes, 10,000 drugs, and 100,000 gene-by-drug interactions, right? So the idea, when you explain it in this way, sounds very simple. Just take the integrated value of influence, sum up the scores that you get when you look where all of these are in the drug-gene database, interaction database, and then you just rank them from highest score to lowest score, right? Sounds very simple. So then you get this drug. Any of the clinicians know this drug? Nope. Fostamatinib, used for idiopathic thrombocytopenia purpura, right? And I've said that enough now, I don't get tongue-tied when I do it. Okay, so never associated with pain before. Okay, we got something we're looking for. Okay. Now it turns out that fostamatinib, which is given orally in human beings, because these are FDA approved drugs, right? This is the whole point of this drug gene database, is to try to get FDA approved drugs. Okay, this is a prodrug. The active metabolite is this molecule called R406. So the ultimate sort of proof that this whole idea that Shahrzad had some intrinsic value is this molecule should be, because the way it works, which I'll tell you about in a minute, should reverse hypersensitivity, right? So, of course, the answer is, I wouldn't tell you the story like this if it didn't work out, okay? Of course. So here you go. This is where everyone in the lab, Janice, everybody's in the lab doing blinding and double blinding and all this stuff. So this is the day when all the transcriptomic analysis was done after the nerve injury. This is an intrathecal injection. Remember, all of the transcriptomics is related to spinal cord. And so what you can see is here's the control. This stuff, R406, is a bear to work with, but they found, they found a solvent that would work, that wouldn't really do anything on its own, but reverses hypersensitivity. And this happens in both males and females as we expected. Okay. Now, the drug was designed to target a very specific enzyme, a tyrosine kinase called SYK. This, for your viewing pleasure, is the crystal structure of SYK, and here's a blow up of the part with R406 in the catalytic domain. So binds to the catalytic domain of SYK and inhibits the kinase. And there's a complicated way this works in ITP, but this is the target. Okay, now, Fiona, was SYK in that list of 93 genes? It was not. Okay, so how the heck did we find that? Okay. So it turns out that the stringency used to put the 93 in that cell in the Venn diagram, the criteria were very, very, very strict because we didn't want to get false positives, right? So when Shahrzad loosened up the criterion just a tad, again, a statistical term, just a tad, then suddenly this network appears, right? You see more genes, but right smack in the middle is SYK, okay? So a little bit of post hoc rationalization and help, okay, to think about how could SYK be there. SYK was there hiding from this highly, highly stringent initial analysis. Okay, so if that's true, then, these are genes that on that side, you saw almost all the genes on those volcano plots went up. SYK is one of them that goes up. So if that's right, then we should be able to independently see changes in SYK mRNA, and we do that in the males and in females. And if it's really, remember I told you I'm a physiologist, SYK protein may follow, or sometimes genes don't follow, or protein doesn't follow transcript levels. But in this case, SYK protein is up in males and females compared to the control. Okay, so now this is like, we're feeling a little bit more confident. The only thing was that I had, my thing about drugs is every drug has two actions, the one you know and the one you don't know. So maybe it was just a fluke, right? Maybe it was a complete and utter fluke that we had pulled this out, everything worked out so brilliantly. So turns out because ITP seems to be a decent market for SYK inhibitors, there are other drugs that have been developed, molecules. And so we got one that seemed to be, relatively soluble. This compound called P505, those of you that know structure can see it's a structurally distinct molecule. It does the same thing, reverses hypersensitivity in males and females. And so now we're feeling much more confident, right? That we've got these two structurally distinct drugs. We've got evidence for SYK going up. It really looks like we've identified this target, which was completely unknown in pain until Shahrzad took this particular approach. So we're now thinking about, well, how does this work? How does it all fit together? Turns out in immunology, SYK is very well known as an adapter protein that is downstream a lot of different kinds of cell surface receptors. So we've got lots of targets. We've got Plex7A mice coming right now, right? So, so we're trying to work through what's going on, what are the signaling pathways that are involved. Can we get inhibition of hypersensitivity by systemic delivery of the SYK inhibitors, not just by intrathecal delivery, and those kinds of questions. But the real question is, where in this cartoon does SYK go, right? Because it either goes somewhere in this cartoon, or we have to make a new cartoon, right? If it doesn't, if we can't figure that out. So we're trying to figure out, which cells might it be in? most likely candidates are immune cells. Which ones? Well, it might be these ones, maybe. Probably not these ones because of the way we did the experiments. My bet is on somewhere over here. But a few neuron types do express SIC, so maybe there. So we're trying to work through that. And the real reason for doing this was try to fast track this FDA approved drug to get it out into patients. So we've been thinking about this. I must admit, I'm still a little hesitant to start to go directly to humans with this SIC because pain clinical trials are so fraught. We got to get it right. So I think we need a little bit more evidence that at least we can reverse SNI-induced hypersensitivity, at least with systemic or oral administration of the drugs before we go. And then, Eder and other people in the lab are going to figure out which cell types and where and all this kind of stuff. So that's where we're at. We still have this, cartoon that sort of drives everything, at least the way that I think about it, at the spinal cord. Pain is in the brain. So this changes that we have in the spinal cord then have to be manifest in changing the brain networks, the nociceptive and pain networks in the brain. How that happens, we're leaving to other people that are much smarter than us, but that's sort of where we are. So this has been a wonderful sets of experiments done by many people in my lab and with our collaborators and with Sharzad and the Center for Computational Medicine and even Dave Finn from Galway. So thank you very much again for the chance to come and talk.
Any questions for Dr. Salter?
So our friend Jeff would say that the problem with using male mice for female patients is not just that they're male mice and female patients, but that they're adolescent mice and rats, and then they're adult humans. So I'm just curious from a developmental perspective, either thinking about sort of maturation or life events, what might look different in this pathway?
Yeah, so these are, I would say, early 20-year-olds, sort of. Okay, so not old people like me who really have a higher incidence of pain, I can tell you from personal experience. But so we haven't broached the question of how this is age-related. This part of it, though, the male part in microglia, Ji Zhang, when she was in Yves de Koning's lab a long time ago, followed animals out for many, many months in SNI, males only at the time. And it turns out that there's changes in the number and the morphology of the microglial cells. But when you do microglial cell ablations or other things, these cells are still, this is in the spare nerve injury model. So that model, there's pain hypersensitivity for as long as the animals live. So G's looked out many, many months. And still, if you do interventions to perturb this side of things, this little quadrant over here, then there's still reversal. So microglia, at least in adult animals, long after the nerve injury, the microglia are still involved. People will tell you that's not true. Tell them G actually did the experiment that they don't know about. Okay, because it's not a well-known experiment, and microglia numbers do fall, and so people correlate. It's correlation between the fall in microglia and that they're not, because there's less number of them, they're not involved. And in fact, G did the experiment to prove that's not true. So, sorry.
Hi, I'm Erin, the pain fellow. Thank you for an excellent talk. We're talking, I suppose, about treatment of pain rather than prevention of pain. So if we think about the layers of things that we can do as a preventative measure, and we know that the brain and the immune system are interlinked, what are the simple strategies that we can do for patients to try and build their immunity to prevent them from chronic pain? My interest is in the gut microbiome, how we can try and optimize that as much as possible in people who already have a lot of stress, oxidative stress, and already a breakdown of that neuroimmune barrier. Does any of your work focus on?
So we haven't focused on that question of microbiome specifically, but there's a lot of people who have and looked at neuroimmune interactions, and Arkady, and I'm blocking on his last name now, at McGill, has a whole story about that, and actually using microbiome replacement in rodents to change pain phenotypes, right? So this is where I think, if we hadn't started to become sensitized to neuron immune interactions, we would have never gotten there. But I think we're getting into sort of clever, different ways of thinking about that, rather than just saying a drug to do this. Or, I told you the story that way about SIC, but there's many ways you can think of intervening in other totally different ways, like microbiome replacement in the gut or other ways to affect immune signaling, which seems to be quite plastic. Right? So,
thank you.
Thanks, Mike.
Yeah, the way these things work is that if you do this thing properly, it goes solid for the speaker, and then whoever's waiting, it flashes, and then when I hang up, the next person goes. It's quite clever. Anyway, I drive a Mini. This question is more kind of pragmatic and not so sciencey, which is if you have an old drug that's licensed, relatively inexpensive, and doesn't cause too much harm, why do you need to wait until you've proven how it works in some subset of animals for some particular indication when you have surgeries going on, particularly I'm thinking of, mastectomy and very high incidence of neuropathic pain after surgery? Why not devise a pragmatic trial that could go on in parallel? Thank you.
Yeah. So I guess this is just me being maybe a little overcautious and trying to think like, what would it take for us to say it's not really a good idea? And I think if these drugs don't work by systemic administration, that would be a game changer for me. And so then, I mean, there are on and off target side effects of these drugs as well. They're not totally benign. So to expose pain patients to that, I think we need a little bit more validation that repeated dosing will do something, that we have this ability with this or any molecule, right, or any therapy really. I mean, no matter how safe it is, like, does it work under the conditions you think get as close to what's happening in the clinic? So then I think if we can get that, I think we're going to be able to have a stronger set of evidence to say to someone who might want to pay for that kind of trial to do that and be on firmer grounding. And I think, Hannah mentioned that there have been 11 trials of glial modulating drugs. They've all been underpowered. They've used horribly crappy drugs. Here's a good drug. It's well targeted. I mean, the primary molecular target is really well known. So I wouldn't want to sort of blow it by going too quickly and getting either the wrong patient population or finding out, oh yeah, when we give this in rodents, it doesn't cross the blood-brain barrier or something like that, right? So we're not quite there yet. There is no blood-brain barrier information. Is there ever on these drugs? No one's looked at that. So we've tried to look at that. So if we get there, the alternative, if it was super safe, you could just say, why don't we just try it, right? Which do take the ethical perspective that would be, a reasonable thing to do if it's safe. Maybe it's just being overcautious, as I said. So.
I'm sorry to ask you a question about immunology when I'm
this
just wondering.
This is the extent of my knowledge.
For the immunological components there that do show sex differences, do we know why those sex differences exist? Are the adaptive immune cells or the microglia, meningeal macrophages, do you think they're part of the pain pathway in different ways in males versus females because of hormonal differences or because of genetic differences, or
so
why?
We've worked that out mostly around this part here. And Shahrzad, we've pretty much nailed what the molecular difference is here. So you see this part sort of starts with, at least in my mind, starts with upregulating P2X4 receptor expression, and then things happen after that. Calcium comes in, P38 macro, blah, 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 right? So it turns out that what Kazu, my partner in crime, had done, there's a whole other part to this cartoon about how does the signal from the injured nerve get to change anything in the spinal cord, right? And so Kazu, we kind of divided the world in half, right? We would take this part here between microglia and neurons, and Kazu would take between the nerve injury and the microglia activation, right? And so he's worked out this beautiful set of pathways that are involved in that. And one of them is a two-step promoter pathway, let's call it, that goes from two factors, IRF5 and IRF8. And it turns out that IRF5 binds to the promoter of the P2X4 receptor gene. There's two binding sites for IRF5. One of them is hypermethylated in females, preventing the binding of IRF5, and so that P2X4 is unable to get upregulated in terms of message expression. So we've got it as close. Why that happens, that's hormonally based, actually. We knew that from the experiments we did with Jeff. It seems to be related to testosterone, so lack of testosterone in females. So that part we've worked out in quite a bit of detail. This part, still don't know totally which adaptive immune cells are involved and how that part works. But I think we've got this part. The rest of it is open. Hannah's working on part of this, probably going to work a little bit more on this, right? So yeah, the hows, that's some of the big questions we're working on. Why is a different question that we have to have beers to talk about.
Thank you so
Thanks.
much for a wonderful talk.